before we leave this figure, let's think about some real parameters, reminding you that this plot here is showing us the degree of saturation, right? The extent of binding of a protein and its ligand, and the 50%, or the KD, the concentration of ligand at which we're 50% bound. For myoglobin, that KD is something like 2.8 torr in terms of the concentration of oxygen, or the partial pressure of oxygen. That is to say, at 2.8 millimeters of mercury, we're 50% saturated. Our myoglobin should be 50% saturated. That compares with something more like 20 to 40 torr in the tissues, the partial pressure of oxygen, pointing out that under standard physiological conditions, the myoglobin should be almost completely saturated, well above the KD. And I think there you can see the power of understanding the KD relative to your experimental parameters or conditions. To put that in perspective, let's look at these numbers that I kind of kludged together for you here. Just thinking about oxygen dissolving in solution, not binding to myoglobin, but dissolving in solution. The concentration of oxygen in water, something about 0.03 millimolar. In the blood, concentration of oxygen is about the same. And in the tissues, we see it's somewhat less, right? Remembering that the concentration in the tissues is going to vary depending on exercise, blah, blah, blah. But the concentration of oxygen is a little bit less, 0.01 millimolar oxygen concentration dissolved in those tissues. Now, keep that in mind when we begin to think about what myoglobin is doing. And we're going to back up a little bit and think a little bit about metabolism here. I think it's important to put the function of these biological molecules, myoglobin and hemoglobin, into context. What's happening with oxygen? Well, we breathe it in to our lungs from the air. The oxygen goes into our blood via hemoglobin and travels throughout our body through capillaries, finally finding its way into muscle tissue, where it has to leave the hemoglobin now and enter the myoglobin. So there's a couple of things happening there, right? There's oxygen going into the solution and going from the solution into the hemoglobin, which we'll discuss in detail in the next series of lectures. And then the hemoglobin's got to give it up, and it's got to diffuse into a cell. The oxygen has to go into the cell and then be taken up by the myoglobin. But the myoglobin has to give it up too. The myoglobin has to give the oxygen up to the mitochondria, remembering that the point of oxygen consumption is delivery of the oxygen to the mitochondria for energy production. Oxygen is the ultimate electron acceptor in mitochondrial electron transport, and we're not going to go deeper into that, but just to say that the end point here is the binding of oxygen by cytochrome C oxidase, and electrons are accepted from electron donors. That's mitochondrial electron transport, but ultimately those electrons go to the oxygen, breaking it up into water. Now, that's the pathway, and what I want you to think about is what's got to happen with the oxygen and the oxygen carrying or binding molecules. So let's think about, and we're skipping ahead a little bit to hemoglobin because you can't really separate the functions of hemoglobin and myoglobin very effectively. But I'm asking you some questions here just to chew on. How does hemoglobin affect blood oxygen concentration? Remembering that I told you that the concentration of oxygen dissolved in the solution is something like 0.01 millimolar. The hemoglobin is about 2 millimolar in your blood, right? And that, because there are 4 oxygen binding sites on a hemoglobin molecule, if you didn't know that yet, you're going to know it by the end of this series of lectures. But that says that the concentration of oxygen, due to hemoglobin binding it up, is 8 millimolar. Okay, remembering that's like a factor of 100 higher in concentration than that which would be in the solution itself. So I want to make that clear to you. There's a point of hemoglobin in the blood, and that is to maximize the amount of oxygen that we can carry to the tissues, well in excess of that which we could dissolve in the solution itself. We got to think though about how we ultimately transmit that oxygen to the myoglobin, and then how we desaturate that myoglobin in the myoblast, well, let's say myofibril, to move it into the mitochondrial electron transport chain. I'm not going to answer that question for you, but I'm going to show you some data that's suggestive of what kind of stuff might happen, and at least allows us to think about the processes. The only thing I will say is that consumption of ATP during exercise, right? That's the point of mitochondrial electron transport is to replenish our ATP. So you can imagine that if this is the rate we're consuming ATP, 3 millimolar per second, remembering what our oxygen concentration is, what we're, how we're going to replenish that ATP is the point of the electron transport chain. The question is, how does exercise affect myoglobin saturation levels? Realizing that in order to replenish that ATP, we've got to take oxygen off the myoglobin and feed it to the mitochondria. So that's the story that I want us to think about. And I have a couple of fairly complicated slides that are going to describe some of the experiments that folks are doing to try to understand this myoglobin saturation and desaturation during exercise. What I'm going to show you are NMR experiments. Again, looking at the flux of myoglobin and the flux of oxygen through myoglobin. And the story begins with the fact that histidine can exist in a bunch of tautomeric states. Here's our molecule of histidine. Here's our side chain of histidine. We know that it's protonated. Okay, well, this is a detail that we never delved too deeply into, did we? When we were beginning to talk about amino acid structure and protein structures. But we can protonate that guy at the delta nitrogen or the epsilon nitrogen. And in fact, we would see both. In free histidine, we do see both. But in free histidine we predominantly protonate at the epsilon position. Well, okay, fine. Free histidine, again, what happens in a real protein differs from what we might expect to observe in solution for a free amino acid. And in fact, in myoglobin, we see exclusively and here we're talking specifically about the proximal histidine, right? Exclusively the protonation of the delta nitrogen. Okay, so we should be seeing, we haven't drawn it here, but this delta nitrogen, we should have a proton dangling off of the end there. And isn't that interesting? Now, I can also say there's more structural details, right? That proton is actually hydrogen bonded to a serine elsewhere in the protein, but that's, the story gets as complicated as you want it to be. The detail for us in terms of interesting NMR experiments is the fact that this iron atom, this iron ion, is about 5 angstroms away from that delta proton, easily within range to have an effect, for example, on NMR chemical shifts. And in fact, the experimental handle, right, the metric of oxygen binding that we have to think about in this experiment is the fact that the delta proton spectrum depends on whether or not oxygen is bound at the iron atom. So iron 3, right, we're in the iron 3 state when oxygen is bound. When oxygen is unbound, right, when we give that oxygen up to the mitochondrial electron transport chain, we go to the deoxy state, that's iron 2, so there's an oxidation state change, and we go into that deoxy state as a paramagnetic state that dramatically affects the proton chemical shift of that delta nitrogen. So again, if it's not clear to you, what we're looking for here is a change in the chemical shift of this proton that reports on whether oxygen is bound at the nitrogen. This is a rat experiment, right? So what I want you to imagine is you've got a big NMR instrument with a big bore. We're not dropping little tubes into the instrument anymore. We have a little, I want you to imagine a little rat running around in a ball inside of the detector of the NMR, and we're measuring its myoglobin spectra. That's in fact what's happening in this case. These are one-dimensional proton spectra as a function of exercise time. Each of these spectra represent a 20-second integration, so we've got to measure the data for 20 seconds before we can get decent spectra. But what we see is after we're resting here, right, and our myoglobin is completely oxygenated, so we are not in the iron 2 state, we're not in the paramagnetic state, and so we see no proton spectrum down at this chemical shift. And after we begin exercising, immediately, boom, we see this peak appear. So that's the chemical shift that I'm talking about when we switch to the iron 2 state, myoglobin becomes deoxygenated, and we see this peak grow in very, very quickly. 20 seconds, 40 seconds, 60 seconds, over several minutes of worth of exercise time for this poor little rat. And what we see, and we're not quantifying these peaks, and at least I'm not showing you that. You feel free to go look at Chung et al. From 2005. What you'll see is that the myoglobin saturation drops to 50%. 50% of saturation. Backing up again, let's look at our cartoon. We should be totally saturated in the tissues, but under some conditions, strenuous exercise, we see that we desaturate. No particular surprise there, right? We know that we're trying to, that myoglobin, its job isn't to hold on to oxygen forever. Its job is to pass along the oxygen to the mitochondrial electron transport chain. So that's just an interesting factoid, I guess, and it points to the fact that we can measure this flux of oxygen through metabolic pathways through quite elegant NMR experiments. And I want to show you one more. Yeah, there's the chemical shift for that proton. This, okay, I'm laughing because the experiment in this case is measuring sleep apnea in seals. And again, these are whole animal NMR experiments. We have an NMR instrument. We don't, but these Ponganis et al. Have an NMR instrument with about a 1 meter bore, and they can fit a seal in there. And the funny thing about seals is that they undergo sleep apnea. What is sleep apnea? Well, some of you may be aware of the fact it's a very common problem in humans, especially in the obese. We see people stopping breathing. Their breathing halts for a certain period of time while they're asleep, and then it starts up again. Well, what do you expect happens when you stop breathing? You stop delivering oxygen to your lungs, which replenishes the oxygen in your blood, which replenishes your oxygen in your myoglobin in your tissues. So of course, what we see in these, and this is time, right? And this poor seal is snoring away, but stops breathing eventually. And what we see is that during the apnea periods, we see this big peak, right, of myoglobin deoxygenation. Oh, he shakes and wakes up and starts breathing again. And then the oxygen concentration, the deoxymyoglobin concentration goes down and disappears. And then we enter another phase of apnea and we see it pop up again. So I think that's just a really interesting, an interesting metric. And the idea in this experiment is this particular group might be interested in understanding what's happening as a seal is diving and holding its breath. They're interested in the metabolism of diving, and it's really interesting that they think they might be able to use apnea as some kind of a model for exercise. Although you could shoot holes in that if you want, and go to read this experimental biology paper if you're interested. But just as a figure, or just a summary of their data. Here we're looking at a plot of oxygen concentration, relative saturation of oxygen as a function of time, and here we see an apnea period, and then these are breaths, right? These are different breaths that the animal is taking, and then we enter another apnea period. This is quite a long period, so you could imagine if you're working with these animals and you see that there's an apnea period of 10 minutes, that's not going to slip by you. So you might think this might be an interesting model for understanding diving, right? Because those animals are also diving for several minutes at a time. In any case, what we see is the saturation of myoglobin drops down. Now we're only seeing it drop down maybe 15%, right? Almost saturated when we're breathing. We stop breathing, and the animal's saturation level drops down low, which is very, very interesting. But it says that that 10 or 15%, that's what's controlling the flux of oxygen to the mitochondrial electron transport chain. But you know what? You don't have to simply look at proton NMR spectra. We have other nuclei that are available to us. And in the same paper, these guys describe a phosphorus NMR experiment where they're monitoring the same thing during here's a pre-apnea period or breathing period, apnea period, breathing period. Here we have longer integration times. You'll notice 70 seconds or so. Okay, so what are we looking at in a phosphorus NMR experiment? Well, you guys probably are well aware where we see phosphorus in metabolism. We all know about ATP, adenosine triphosphate. We all think of that as the energetic currency of most living organisms, right? And the question that we're asking here is, well, what's happening to the ATP concentration? And more importantly, is there something happening to the phosphocreatine concentration? And these if we're undergoing major fluxes in metabolic fluxes and changes in metabolic rates, we should be seeing ATP go away, phosphocreatine concentrations creeping up. And the bottom line is that even though they're seeing this up and down in terms of oxygen concentration in myoglobin and a drastic drop in oxygen, well, 15%, a drop in oxygen concentration on the myoglobin, we don't see any real changes in the metabolic processes, right? Electron transport is not affected. The concentration, the relative concentrations of ATP and phosphocreatine are not changing. So I don't want to present that as being too confusing, but just to say that that there's a dynamic process at work here between balancing oxygen concentration in the blood and then in the tissues and then in the mitochondria itself. So yes, we can watch the myoglobin concentration, the oxymyoglobin concentration bounce up and down as oxygen is available from the blood, but that doesn't necessarily translate to large-scale radical changes in metabolism, which we would hope would be the case, right? We want to be able to exercise, and during exercise, we want to be able to maintain a more or less constant metabolic rate, or else we'd fall over and die when we exercise. So that's not an acceptable outcome. The point being, however, that we have these spectroscopic tools that we can use to monitor changes in oxygen concentration in myoglobin. So I hope that was valuable to you. What I want to return to now is a discussion of myoglobin structure. And we'll move from myoglobin structure into a discussion of hemoglobin structure. So we like models. Let's look now at hemoglobin, starting with the fact, as we've already discussed in our evolution discussions or talks, that the two have remarkably similar tertiary structures. So what I'm overlaying here, I believe what I have done is overlay alpha and beta hemoglobin on the left. So obviously alpha and beta hemoglobin are very similar to one another. And then I've overlaid myoglobin and alpha and beta hemoglobin on the same structure, and you see again that they're virtually identical in terms of not only their secondary structural organization, but the relative placement of the heme. Remarkably similar tertiary structures. No surprise there, since they're relatively well-conserved molecules. Even though they are well-conserved, and we can look for that conservation in terms of placing the iron and oxygen binding determinants. Even though they're well-conserved, we can still look at the differences and make and establish a relative hierarchy, if you will, of evolution, right? So that is to say, the hemoglobins emerge later from simpler systems, and that makes perfect sense. Myoglobin, the simple oxygen binding or oxygen storage system, giving rise to something that's much more complicated, that is hemoglobin. Okay. That hemoglobin, tetrameric molecule, so 2 beta subunits, there I have them in sort of this magenta, 2 alpha subunits sort of in this bluish gray. We consider hemoglobin to be a tetramer, but we also consider it to be a dimer of dimers. So we've got an alpha 1 beta 1 dimer and an alpha 2 beta 2 dimer. You got 4 monomers here, each with their heme. You can see the hemes here, 1, 2, 3, 4 hemes. This serves as a mammalian oxygen transport system. Obviously a much more complicated system than the monomeric myoglobin. What's the point of such a complicated